Is it visible?
Yes, sir. Visible, sir.
Okay. So this was a presentation I made long back for. This is what Dr. Ammo said. Heme means blood, stasis means stop. But it is a process in which a series of reactions occur among coagulation proteins to finally end up in conversion of soluble fibrinogen into insoluble fibrin strands. This is the full definition. So this is how, like a pack of cards falling down, you get this sort of reactions going on. And purpose of coagulation is to maintain a balance between pro and anticoagulant pathways. So it should always be balancing between each other. And the main three components, as she rightly said, is the vascular endothelium, which is the first and foremost thing, platelets, and plasma clotting factors. These are the three components which are very, very important for coagulation to take place normally. Vascular endothelium, platelet, and plasma clotting factors. Now, there is a mnemonic to remember. Of course, you may remember the mnemonic, but not all the names all the time. Anyway, freshers party tonight. Come, let's sing and also call seniors. Please have fun. So these are all the first letters. If you take, you can remember that. Coming to the vascular endothelium, when it is healthy and intact, it is antithrombotic. You don't want any thrombus formation in a healthy vessel in a normally flowing blood. So there are mechanisms to prevent, because all the clotting factors and the platelets are all there in the blood all the time, and they are in an inactive form. And even if there is a slight rushing with the endothelial surface, it can get stimulated. So there are anti-thrombotic factors which are in the form of heparan sulfate, um, thrombomodulin, CD39, all these things, prostaglandin, nitric oxide, all these things are all there to prevent a thrombus formation. But and the antiplatelet effect is due to the negative charge repelling platelets, just like in the kidneys where albumin is prevented from getting filtered by the negative charge in the basement membrane and the glomerular structures. There is negative charge repelling per platelets by synthesis of prostacyclin nitric oxide and ADPase. And anticoagulation effect by expression of inhibitors of coagulation like thrombomodulin, which involves protein C and S, heparan sulfate, tissue factor platelet inhibitor, and TPA inhibitor. All these things help in preventing a thrombus formation in an intact vascular endothelium. Whereas when there is a damage occurring, it becomes prothrombotic. How does it become prothrombotic? Because there is an exposure of extracellular matrix, and subendothelial space has got collagen, von Willebrand factor, and platel adhesion glycoprotein. And tissue factor, which initiates coagulation cascade, is expressed from the fibroblast and the extra cellular matrix. So this is the extrinsic pathway that is outside the lumen of the blood vessel. Why it is called extrinsic pathway? Because this pathway is outside the lumen of the blood vessel, but in the subendothelial matrix area. So it is not in the lumen. And the platelets characteristics are they are derived from megakaryocytes. They have a lifespan of 4 to 7 days. They are non-nucleated. That is very, very important. And its membrane is highly reactive when it comes into contact with the extracellular matrix. And once it comes in contact with it, it undergoes adhesion, activation, and aggregation. These are the three processes that undergo. So adhesion is the sticking of platelet to the subendothelial matrix. And this is the primary plug which is done, but they are loosely attached. They cannot stay there effectively if they are left like that. So this is called the primary plug or hemostasis. Once they get attached there, they are immediately activated by the von Willebrand factor, and they undergo what is called degranulation. There are two types of storage granules inside the platelet, alpha and delta granules. The alpha granules release further fibrinogen and von Willebrand factor, factors 5 and factor 7, platelet derived growth factor, all these things are released from that. And delta granules release ADP, ATP, calcium, 5-hydroxytryptamine, histamine, epinephrine. All these things are released by the delta granules. And once they are released, further platelet aggregation, that is addition of or sticking of platelet to platelet, is called aggregation. So the tissue factor release, phospholipid complex expression, thrombin activation, fibrin proliferation, all these things lead to the secondary hemostasis. So the further aggregation of platelets and their stabilization is what is called the secondary hemostasis or plug formation. So these are all the plasma coagulation factors. And coagulation, this is the classic pathway. Extrinsic pathway, intrinsic pathway, and final common pathway is called the classical pathway. So these are all the steps in extrinsic pathway. First initiates the whole thing, then it leads on to the intrinsic pathway, and ultimately that results in activation of factor 10. So both end up in both the extrinsic and intrinsic pathways end up in activating factor 10, which stimulates factor 5. And to make it, A means activated. When it is written without A, it is idle or sleeping. And this is the final common pathway where prothrombin gets converted to thrombin, which converts fibrinogen to fibrin. This is the ultimate final step. And the factor 13A helps in cross-linking of the fibrin clot to make it stronger. And this is a very simplified picture. Injury causes the release of tissue factor and factor 7A, which is the extrinsic kinase, and it stimulates this intrinsic pathway, and then finally leads to the stimulation of factor 10A. From there, it all goes into that. And what are the pitfalls? Why we have to think of some other method of description is it is described using in vitro, that is test tube method. These were all done in the laboratory, and this is what you found out. It failed to reveal the role of factor 7 in the integration of extrinsic and intrinsic pathway in body. It also explains offers no explanation how deficiency of factor 12 does not cause any undue bleeding. Even though you say that factor 12 is involved in the intrinsic pathway, if you have factor 12, you won't have much bleeding problem. And why in hemophiliac there is no support by the thrombin form to control the bleeding? All these things were not explained. So there was another concept mooted called cell-mediated pathway. This was proposed by Marine Hoffman, the lady, in 2001, and all reactions are said to take place in the surface of two different types of cells. One is the platelets and the cells with the tissue factor on their surface, like fibroblast, smooth muscle cells, and it occurs in three phases which overlap each other. That is what they describe. Instead of saying that the extrinsic pathway stimulating the intrinsic pathway and then ending up with a common pathway, Marine Hoffman said it is not like that. You have overlapping of all these things. So if you see here, there is an initiation, which is the as we saw in extrinsic pathway, tissue factor and factor 7A causing conversion of factor 10 into factor 10A. At the same time, this also activates factor 5 and 5A, which also activates part of the 10A, and then whole thing goes into that. And then there is amplification and propagation. So what she said is, it is not taking place at time intervals. All of them are going simultaneously, and you get these things. So by this, she was able to explain that why some people bleed and why some people are not bleeding. So initiation is the first phase, and the amplification and propagation are the next phase, which completes the formation of fibrin. So this is a very easy way to remember the steps of coagulation. Step 1, you have to write that target factor 10. So put an X there. Second step is write the numbers from 12 to 8. Exclude 10 since it is there already as your target. So 12, 11, 9, 8, you can put them. Step 3, write 7 for the extrinsic pathway, right? And step 4, draw the common pathway, 1 into 2 into 5. So these 5 factors are involved, and your coagulation steps are complete. Now applied physiology, Von Willebrand factor deficiency causes bleeding due to failure of primary hemostasis. Platelet can be deficient in number or its function, which can also lead to failure of clot formation, resulting in excessive blood loss. Plasma coagulation factor deficiency results in failure of progression of coagulation cascade, leading to blood loss. So these are the three important mechanisms by which you can have a bleeding tendency. And failure of anticoagulant factors, sometimes the excess of procoagulant factors can also lead to thrombus formation in the arterial or venous system. And deficiency of antithrombin 3, protein C, protein S are the common causes to promote abnormal intravascular coagulation. And excess presence of antiphospholipid antibodies and homocysteine may also promote thrombosis. Rarely, primary thrombocytopenia, where the platelet count is abnormally high, can also cause spontaneous thrombosis. So these are all the factors resulting in intravascular thrombosis. So evaluation, you can do it by history taking, lab-based tests, and point-of-care testing. And these are all things which are, of course, not really related to the talk I gave was on both physiology of coagulation and the test to identify coagulation deficiencies. So just rush through that. They're all the lab-based coagulation tests. Point of care testing, nowadays we have activated clotting time, which is commonly used in the cardiac OTs. So all of you worked in cardiac theatre will know, before allowing the surgeon to cannulate the vessels, you give heparin to the ACT, and then once you get the satisfactory activity of heparin, then only you allow the surgeon. Then high dose thrombin time. And viscoelastic measure, thromboelastography is very, very commonly used in the exam also. And the rotation rotum is there, then sonoclot.
This can be a complete short notes, sir. This can be a complete short notes for them.
And clotting
Point of
factor
care
test.
testing.
Vascular endothelium, platelet coagulation factors are the three backbones of coagulation: vascular endothelium, platelets, and coagulation factors. And there should be a balance between pro and anticoagulant factors, which will result in bleeding or thrombosis. So cell-based pathway seems to be a correct way to describe coagulation and likely to replace the classical pathway from textbooks sooner. Diligent history taking is what is needed in majority of patients to predict bleeding or thrombotic tendency. Time is not far away for point of care test to replace traditional lab-based tests. Okay.
Thank you, sir. Thank you very much, sir. It was very, very capturing, mind-blowing presentation, sir. It has made, and especially the mnemonic-based slide was really very appealing for all the students, sir. In one slide, they can remember all the classical pathways so easily with numbers. 10, 12, 11, 9, 8, this side 7, bottom line 1 to 5. That's all.
Okay.
Just remember a Y.
Actually, this point of care testing can be short notes by itself.
Yeah.
Yes, sir.
And
Yes,
what
sir.
sir has put all
Not
the
just,
latest also. He just
will
put
be
in all the
useful.
latest also. That will be very useful.
I know. I think
Any
ஒருத்தர் காதலையும் ரத்தம் வரலையா வடிவேல் கேஸ் மாதிரி been talking for so long. Okay. Any questions or any doubts? We can
Any questions from the students? Sir, what shall we have for the next class, sir? Anything to make interesting? Dr. Amu wants to ask something?
Anything else okay, sir? Which topics are, sir?
We will prepare questions like this and circulate. Okay.
Yes, sir. Okay, sir. Can we have ASCII also, sir, to make it interesting? 1 or 2 ASCIIs?
Then we'll have to cut down on the theory questions.
Yes, sir. Okay.
So maybe 4 questions and 2 OSCEs. Complete discussion will be good, sir.
Okay. We'll do that.
Yes, sir. 4 or 5 questions and 2
most
ASCIIs.
confusing or difficult questions, and then go for ASCII.
Yes, sir. Okay, sir. Okay, sir. If no more questions are to be discussed or doubts from the students, can we sign off this meeting, sir?
Sure.
Yes, sir. Thank you, sir. Thank you, madam. Thank you,
Thank
dear
you,
students.
Vasanthi. Thank you very much for the contribution.
We'll meet next week and once again next Tuesday. Thank you, sir. Good night, sir.
Thank you, sir.
Thank you, madam.